In this module, I will be helping you to revise the topic of how substances are transported in plants. We shall need to revise the structure of roots and how they are adapted for their function, also the uptake of water and ions from the soil, the roles of transpiration, root pressure and cohesion tension in moving water through the xylem, the adaptations of xerophytic plants, that's plants that live in very dry conditions, the structure of stems, translocation of organic substances in phloem tissue, and experimental evidence. Let's think about the structure of a plant. The roots anchor the plant in the soil and take up water and mineral ions. The stem carries water and minerals to the leaves and also carries organic material made by the leaves to roots or other non-photosynthesizing tissue, such as buds and flowers. At the root tips, the epidermis is modified to produce a pyliferous layer. Here, the cells have outgrowths called root hairs. This gives an increased surface area. Mineral ions are taken into the root hairs by active transport. This lowers the water potential inside the root hair cells, and water from the soil enters the root hairs by osmosis, down the water potential gradient. This raises the water potential of the root hair cell, so water passes to the adjacent parenchyma cell in the cortex by osmosis. This cell now has a higher water potential than the one next to it, so water passes into that cell by osmosis, down the water potential gradient. Water can pass through the walls of the cells and from cell wall to cell wall through the intercellular spaces. This is the apoplast route. It may pass through the partially permeable plasma membrane and into the cytoplasm, and through the tonoplast membrane into the vacuole, and on through the cytoplasm and into the next cell through the wall and plasma membrane, or through the plasmodesmata. This is the symplast route. In the centre of the root is the stele. This is where the vascular tissue, xylem and phloem is situated. There is also cambium tissue. This is meristematic. The cells can divide to produce new xylem and phloem tissue. The central positioning of the stele makes the root resistant to the upward pulling forces. Surrounding the stele is a layer of cells, endodermis cells, that have a waxy waterproof band of suberin in their cell walls. This band is called the Casparian strip and is impermeable to water. It shuts off the apoplast pathway so that water has to pass through the cell membranes via the symplast pathway. Nitrates in the water are actively passed into the xylem vessels, lowering the water potential in them so that water will follow by osmosis. As the membrane proteins needed to actively load the nitrates are in the plasma membrane, shutting off the apoplast pathway ensures that the nitrates can be passed via the plasma membrane proteins. In older roots, many of the endodermis cells are completely suberized, which means that both apoplast and symplast pathways are shut off in these cells. A few cells called passage cells remain with just a band of suberin, and these allow water to pass through by the symplast route. At the leaves, water is always being lost by transpiration from the stomata, most of which are on the underside of the leaves. Stomata have to be open for gaseous exchange, and inevitably, water vapour will be lost from them. Water evaporates from the cellulose walls of the leaf cells into the air spaces in the mesophyll layer. Water from the xylem in the vascular bundles in the leaf moves into the mesophyll cells of the leaf, and this reduces the hydrostatic pressure, or causes tension at the top of the xylem columns. Water passing into the roots causes a higher hydrostatic pressure at the base of the stem. The tension from above, root pressure from below, and high cohesion means that water moves in a continuous stream up the xylem vessels. This is known as mass flow. There is also adhesion produced by attractive forces between water molecules and the inside surfaces of the lignified xylem vessels. The plant uses none of its energy in this process. Younger xylem vessels have spiral bands of lignin, whereas older vessels have walls heavily impregnated with lignin with many pits. Each pit is an area where no lignin was deposited, so water can move sideways from one vessel to another through the cellulose cell walls. Xylem vessels consist of many xylem vessel elements arranged end to end, whose end walls have broken down. These vessels or vessel elements should not be called cells as they are non-living. The vessels have hollow tubes of small diameter so that water can pass through them. They are strengthened with lignin to prevent their inward collapse. Notice that the vascular bundles consisting of phloem, cambium and xylem are arranged around the outside. This gives mechanical support to the stem. A potometer can be used to measure the rate at which plants take up water. If we assume that they lose water at the same rate that they take it up, we can compare rates of transpiration in plants under different environmental conditions. The stems have to be cut under water to avoid air entering the xylem vessels. The whole apparatus has to be assembled under water, but no water must touch the leaves. Greased rubber tubing tightened with thin wire prevents leakage of air into the apparatus. An air bubble is introduced into the capillary tube, and the movement of this bubble, measured against a scale, gives an indication of water uptake in a certain time. The apparatus is allowed to acclimatise to the conditions, and then the distance travelled by the air bubble is measured. The reservoir tap is opened to reposition the air bubble, and the procedure is repeated twice more to obtain an average reading. If we know the diameter of the inner bore of the capillary tubing, we can calculate the volume of water taken up by a leafy shoot per minute. The volume is calculated by pi r squared d, where r is the radius of the inner bore of the capillary tube, and d is the distance travelled by the air bubble in 1 minute. If we find the area of one of the leaves by drawing around it on graph paper, and then count the leaves on the shoot, we can estimate the rate of water loss per unit area of leaf per minute. This enables us to accurately compare water loss from different shoots. If we simulate windy conditions, then we find that the rate of transpiration increases, as it does in bright light and in warmer or drier air. Wind removes the water vapour from around the leaf and maintains a steep water potential gradient. Light causes stomata to open. Dry air increases diffusion of the evaporated water from the leaf, as dry air can absorb more water vapour. Some plants, such as marram grass found on sand dunes and cacti, are well adapted to living in dry areas. Such plants are called xerophytes, and the adaptations include having rolled leaves so that the water vapour escaping from stomata is not able to diffuse away. This means that the stomata open onto a humid space and the water potential gradient is not steep. Hairs, such as those found on geranium leaves, help to trap a layer of moist air next to the leaf's surface. Cacti have very reduced leaves and flat, swollen stems that store water. The leaves are reduced to spines, and these reduce the surface area from which transpiration can take place. Translocation. This is the term used to describe the transport of assimilates, soluble organic substances made by photosynthesis. Assimilates are translocated in the sieve elements of phloem tissue. Sieve elements are joined end to end to form sieve tubes. Each sieve element is a living cell with some organelles, but with very little cytoplasm, no nucleus and no ribosomes. Where the end walls of two sieve elements meet, a sieve plate is formed. Each sieve plate consists of the two end walls perforated by large pores. Each sieve element has a very metabolically active companion cell. There are many plasmodesmata between each sieve tube element and its companion cell. The sucrose produced at leaves, called sources, is actively loaded into sieve elements. This lowers the water potential in the sieve elements, and so water enters by osmosis down the water potential gradient. At the roots, or sinks, sucrose moves out of the sieve elements to be used for respiration, or to be converted to starch for storage. Water follows by osmosis. Thus, at the sinks there is a lower hydrostatic pressure than at sources. so the phloem sap travels by mass flow down the pressure gradient. Some sinks, such as buds or flowers, are above the sources, so phloem sap can move up and down the plant, unlike water in xylem, which only moves up. You need to be aware of the experimental evidence for phloem as the tissue where translocation takes place. This evidence includes experiments on ringing, use of radioactive carbon, electron microscopy, use of aphids to obtain sap that is subsequently analysed, and use of respiratory inhibitors. Early experiments involved removing a ring of bark from a tree. This removed the phloem tissue but left the xylem intact. After several weeks, the tissue above the ring was swollen, showing an increased concentration of solute, indicating that removing the phloem prevented these assimilates from passing from leaves above the ringed area to areas below. Mature leaves were exposed to radioactively labelled carbon dioxide, where the carbon atoms contain 2 extra neutrons. Such atoms are unstable and produce radioactivity that can produce an image on a photographic plate. When such an autoradiograph is produced, the radioactivity is largely confined to the area of the stem where the phloem tissue is. If we cut into phloem tissue to collect sap, the sieve elements respond by producing callose to form a clot, so the flow stops. Aphids feed on plant sap by inserting their fine needle-like mouthpart or stylet into the phloem. A feeding aphid can be anaesthetised and its body removed. The mouthparts remain in place and sap from the sieve elements is exuded and can be analysed. The sap can be analysed at different times of the day. This table shows the composition of phloem sap. During the daylight hours, the concentration of sucrose would increase as the plant has been photosynthesizing. Another piece of evidence is that when respiratory inhibitors such as cyanides are applied to phloem, the translocation of organic solutes reduces or stops. If these poisons are applied to xylem, there is no change to the translocation of organic solutes. There is also evidence that phloem transport occurs by mass flow. Firstly, that the rate of transport in phloem is about 10,000 times faster than if substances were moving by diffusion alone. Secondly, that the actual rates of transport measured coincide closely with the rates calculated from measured pressure differences at sources and sinks. There is evidence that sucrose is actively loaded into phloem. Phloem sap also has a relatively high pH of about 8, consistent with hydrogen ions being pumped out of the cells. Also, the potential difference across the plasma membranes of these cells is minus 150 millivolts on the inside, also consistent with hydrogen ions being pumped out. Lastly, there is much ATP in the phloem companion cells. The hydrogen pumped out of the companion cells will diffuse back in through symport channels, dragging sucrose with it. The increased concentration of sucrose in the companion cell will then lead to its diffusion into the sieve element.